have to start. So we are very happy to have Romana Schirhagl here today, giving a talk to us about nanoscale free radical detection in living cells using diamond magnetometry. It's actually something that we are in DIMA's group very interested in. Romana is Austrian. She was born in Linz in 1984, and she also studied in Vienna, where she did her master's and also her PhD thesis in 2009. Then she went and traveled around to see the world, to Stanford and to ETH as a postdoc until she got a professorship in Groningen in the Netherlands in 2014. So we are super happy to have you here and very interested in your talk. Welcome.
Thank you very much, Arne, and thank you all for being here today and for the kind introduction and for having me, of course. Yeah, I will, as Arne mentioned, show you a bit about my work of the last few years, or at least the end result where it led so far. Yeah, so first let me tell you a bit why we do what we do. So in biology, there is a challenge, and that is measuring free radicals in cells. And why would anyone want to do that? Well, radicals are very important for a lot of different reasons. So first of all, there is a lot of processes where they have a function and where they are good. Like for instance, if you have cell signaling, how they communicate with each other, if you have cell maturation, how they develop. But then there's also processes, and most of the processes where we hear about radicals, they are not the good guys. So they are, for instance, involved in aging. So one of the reasons why people think we age is that radicals are formed while we do our metabolism and then destroy all kinds of molecules in their surrounding, and this is seen as a bad thing. And then they are also involved in all kinds of diseases, and this includes diseases that cause the most deaths worldwide, and for instance, cancer, cardiovascular diseases, or viral or bacterial infections. So they are kind of everywhere, and often they are also seen as a sign that something is going wrong in a cell. So if a cell is dying or something like that, you also have a lot of radical generation. But what's a bit difficult for the state of the art is that they are very short-lived and reactive, which makes them difficult to detect. And as a result, they also occur in relatively low concentrations, which makes it difficult to do anything with them or to use them for diagnostic purposes or things like that. There are currently a few techniques that can tell you something about radicals, but at the moment, there is not really a good method to map them and to see where they are exactly. And you cannot differentiate them very well either. So one technique that is there, it's called qPCR. So there is a whole family of techniques that would go in this category. So what essentially these kind of methods do is that they detect a response of the cell towards the radicals. So they don't measure the radicals themselves, but they measure, for instance, certain RNA that is expressed, or they measure certain proteins that are built. This can be, for example, enzymes that degrade radicals. So if there is an overload of radicals, the cells will respond by creating enzymes that degrade them, and then you can detect this. You can also detect the damage that they do, so you can detect all kinds of damaged molecules. But these methods don't offer you any spatial resolution. You take your whole sample, you lyse it, and then you get, in the end, some information. And the other problem with these kind of techniques is that you need to know very well what you're measuring. So what radicals cause what pathway, and only if you know all these things, you can then at the end do this kind of measurements. On the plus side, they are very specific, so that's nice. Then there is MRI. Just MRI as it is, you wouldn't see anything in terms of radicals because it's not sensitive enough, and it's, yeah, the voxel size that you get is very big, so you just don't have enough radicals in that volumes that you would see something. There's something that people do with MRI, so there's spin labels. You can have a molecule in there, it works a bit like a dye, and it reacts with your radicals and then forms a radical that is stable. Similar thing you can do with optical microscopy. You can have a molecule that reacts with your radical and then forms a stable molecule. The problem with this is that you only get the history of the sample, because these are reactions that are irreversible. So you detect everything that has been built between adding the compound and doing the measurements rather than what you currently have. And since these molecules can diffuse freely wherever they want, you also lose the spatial information to a very large extent. So what we are doing with NV centers is a bit a mixture of the two, right? So we have MRI kind of signals, and we use fluorescence as a readout. So now let me briefly show you what we do with our diamonds. We use a technique called relaxometry. I guess it's familiar to most people in this audience, so I'm not going to say too much about it, but just very shortly. So here on the left, we see an energy diagram of an NV center in diamond. And this is a very special defect in diamond, which can more or less convert a magnetic signal into an optical signal. So what do? We have our diamond, we shine a green laser on it, and as a result, we can pump in this magnetic zero state here of the ground state. And this state is brighter than the equilibrium state. So we shine a laser, and as a result, our diamonds get bright. And this you can see on the right here. At the beginning, at time 0, where we just made them bright, yeah, they are of course still bright. And then we wait for a certain amount of time and probe again. And then we check whether the diamond is still bright or if it has lost this state already. Now, the NV center can only lose this bright state if there is something to interact with there. And so the time that this takes, this decay here, is a measure for the spin noise in the surrounding. And the radicals that I mentioned before, they are creating a spin noise. So in presence of gadolinium, you see the red curve. Gadolinium is a contrast agent in MRI. So this makes a sharp decay. And the blue curve is if you more or less leave the sensor alone. And yeah, you can see that there's very different times that result from this. And this is what we use for radical sensing. So what diamond materials do we use? So while many physics groups are working with single defects and single NV centers, we don't do that. We work with large ensembles, and we use this 70 nanometer fluorescent nanodiamonds from Adamas nanotechnology. They are made by HPHD synthesis, and they contain about 3 ppm of NV centers. So 70 nanometer particles, they have some hundreds of NV centers. So why do we do that? What you lose if you do that is there's a lot of fancy physics that you cannot easily do with these ensembles. But on the plus side, every measurement that we do is already essentially an average of a few hundred measurements. And this is what we need in terms of having the reproducibility. If you would do that with single NV centers, and you have one single NV center that has one property and then the next one is completely different, then it would be very difficult to compare. And with using these large ensembles, we have a bit of a way to get around the poor reproducibility that you get from a single defect being close to the sensor or far away from the surface or in a different nanoscale environment. And by using these large ensembles, we have a way to get a bit around that.
Romana, quick question. These nanodiamonds, they are irradiated and then annealed, right?
Yes, so they are, with electron annealing, I believe, and high doses, and they have, and then they anneal them, at high temperatures. But they are commercially available, so those are what the company does. So we get them already with some hundreds of NV centers, and then we use this for our experiment. So a reason why most physicists don't need to go to this solution, if you can reuse your sensor, and many physics groups do that, they find the perfect NV center and then they work with it forever. Like Jörg Wachtrup at some point said he made 3 Nature papers with this NV center. But the problem for us is when the cell dies and the experiment ends, we have to throw it away. So we cannot reuse the sensor. So this is why we have to live with this, let's say, suboptimal NV centers, because then we can make sure that we can use more of them. So the next thing that we wanted to test is whether these NV center containing diamonds are not influencing the biology that we would like to measure. And we did quite a study on that. And essentially the short answer is no, they don't reduce any of the metabolic functioning that you can see here. So essentially everything that is in the gray bar means they have normal viability. And everything below where you can see only the bars for the controls, those are not doing well. And then we tried all kinds of different diamond particles. We tried different concentrations, timings, sizes, shapes. And essentially, we don't see anything that would alarm us, which is good news. This is an experiment. It's a so-called DCFDA assay. And what this does is that it measures oxidative stress. So this is one of these organic dyes that I mentioned earlier that react with reactive oxygen species. It's not selective for radicals. It measures also all kinds of other species, but it's essentially if you have a high bar here, it means the cells are stressed. And again, we only see high bars for the control samples that have been treated with hydrogen peroxide. So that is also good news. And now I would like to show you a few directions where my group is currently working on. So in the center of everything is diamond magnetometry. We use it in a lot of different ways. And then there are currently several projects where we are investigating. And so if I just start somewhere here, we have a project where we try to show that you can do drug testing. So if you have a drug that is creating a radical response or reducing a radical response, then you can test the functioning of the drug with this technique. We have a paper where we were studying cellular aging. So it's a work here where we are trying to look into this free radical theory of aging. So it's a theory that says that radicals are the main culprit of aging. So what we did there is that we are comparing cells that have aged under different conditions. And then we are trying to see if certain antioxidant treatment, for instance, help in the aging process or not. Then we look into immune responses. I will show more on this later on in more detail. We have a project on viruses, the sperms and fertility I will also touch shortly. Yeah, we also have a project with bacteria, and the diagnosis I will also show you later on. The very short version of it.
Super quick question. Why do you say magnetometry? It seems like relaxometry that you described is a very nice method, but it is not magnetometry, right?
Well, I mean, I called it here magnetometry. We also do a bit different things. We mostly do relaxometry. Yeah, I don't know, if you don't want to call relaxometry magnetometry, then you could also replace by relaxometry here. We also have some things where we measure magnetic resonances. So if you want to call that magnetometry, then you can leave it. But yeah, it's mostly relaxometry indeed. Okay, so now I would like to show you how we get from a relaxometry result from a T1 value to a concentration, because in the end, biologists are not particularly interested in T1 values, but they want to know how many radicals do I have. So what we did there, we need a kind of calibration. So we need something that we can tell how many radicals cause what T1. And now this is not as easy as it sounds, because you cannot just buy some radicals and then you measure them, but you have to make them in a reaction and then measure them. And there's a certain compound, and it's called nono-8. You can also do that with other kinds of radicals. That's just one example how you can make it. But this is a compound that decomposes over time. If you put it in a certain medium, it emits nitric oxide radicals, and then you can measure those. And what we did here is we started with this nono-8, and then we create a known concentration of radicals, and then we measure our T1 response. And then you can see roughly how much radicals you get for what T1. Then we were also thinking of other factors that might play a role. So we tested different things that might influence T1 that have nothing to do with radical formation. So first of all, the temperature. We tried different temperature ranges, and we did not really see any big changes on the T1 at all. So that is good news. We were looking into pH. pH does have an effect. So this is something to be careful about. I think the reason why we see differences in terms of pH is that pH can influence dangling bonds on the surface. So if you have a lot of dangling bonds on the surface and they react differently at a different pH, then you of course see a change in T1. And another thing that pH can cause is that you have some, essentially conductivity or electric currents from ions that move around and things like that. So that can in principle also cause a change in T1, and we see that as well. But in the range where we are, we don't see any significant differences, but you really have to be careful about that. If you change pH drastically, then yeah, I think you would have to do a calibration in the medium at the pH that you're interested in again. So you then cannot use the calibration that we did, for instance.
Anna, excuse me, just a quick question, and maybe you are going to talk about it, but your experimental setup, is it just like some solution and you sprinkle a concentration of nanodiamonds in it and you shine a green laser on it and measure?
For the, well, we have measurements of particles alone. So there we have the particles sitting on the dish, and then we add a solution. So the sample holder holds a Petri dish, and in the Petri dish you can put in cells or biological molecules, or basically whatever you can put in a Petri dish. So it's these special Petri dishes that have a glass bottom. So the Petri dish is already, yeah, sitting on the instrument. And around it is an incubator, so we can control humidity and the gas concentration and stuff like that. Yeah, that's primarily to keep the cells as alive as possible.
May I ask another question?
Yes, please.
So on the previous slide, I'm not sure I quite understood. So you have in green and in blue, what does it mean? And how exactly you control this reaction and amount of nitric oxide? Yeah,
so this is something that the component, you can buy it and it decomposes. And this is something they also use in EPR, for instance, as a standard. So then what you can find is, so you know the decay rate, and then from that you can calculate the concentration. So the way you control it, so the green and the blue are different starting concentrations. And then depending on where you start, you get different, different concentration in the end.
But basically, you don't have one-to-one correspondence, right? You have, if you measure T1 of, let's say, 200, it could be that you have half a micromolar or 3 micromolar.
In the larger concentrations, it becomes, it more or less saturates. So it works more or less, I would say, in the nanomolar range. If the concentration is above that, it's saturated. So
an error is estimated from several measurements, or how do you estimate?
So these are multiple measurements, I think like 9 or so, and then you get an error from the different measurements. And
thank you.
So it's different measurements with different particles. It's also, you have to add the compound, but then you cannot really mix well because it's nanoparticles, and then you will lose your particle. So this makes an error. So actually you get quite an error from the estimation here, because it's quite difficult to make the radicals in a controlled way. So if in one measurement the radical is built, or you add the molecules a bit further away from your sensor, so then this all contributes to the error.
Thank you.
Yeah, so and the last thing that we looked here at is viscosity. So to try if that has an effect at all, we tried different concentrations of glycerol. And basically we don't really see any differences from that. So we have a like 100% glycerol solution and 70% glycerol solution, so that the 100% glycerol is really, really viscous. And we more or less get the same, the same results from those. And then another challenge is, how do you get a nanodiamond in a cell? And depending on the application, that can be different. So depending on the cell type, there are some cell types that readily ingest particles. So one of these cell type is, for instance, macrophages, which you can see in the video on the right. So these are immune cells. If you provide them with any kind of particle, they will take them up. And on the bottom right, there's also an interesting one. So there is a cell that grabs a particle from somewhere and catches it. But what you can also see from this video, by the way, the diamonds are the bright dots here. What's kind of interesting is that these are all the same cells in principle. This is like a monotonous cell culture, but they behave quite differently. So some of them move quickly, some move very slowly or barely at all. So this is interesting to know, and also, yeah, kind of important later on for the measurements.
Romana, why would it reach out and grab something? It thinks it's food or what? I mean, why does it do that?
Or I mean, macrophages, their function in biology is to get rid of particles. So everything they think it's a virus they ingest, or everything that is foreign and doesn't belong here in the body, they grab, and then the body can get rid of that. So amazing. They are quite good eaters. So for those kind of cells, you can fill them up with nanodiamonds until they are whole bright red ball. so I think it's part of their biological function. They just eat everything that is not their own body.
Do you form emotional bonds with some of them?
Sorry?
Do you form emotional bonds with some of them? I find the guy in the bottom here quite cute.
Yeah,
but it's also this particle is very far, so it must sense it somehow to make
that is super interesting that it does that. I don't know why. Also, I mean, if you look at the other cells, they don't do that, right? I mean, there's some that share something on the left, and but like this special grabbing, only this one does it. So with this one, I have an emotional bond. It's a special one. Yeah, so then for this specific cell type, so this is again macrophages that we looked at. The paper where we did this is mentioned on the bottom. That was recently published. And what we did here is we are looking at the number of particles that are in a cell. And I mentioned already that this is a very good eater. So if you plot like time versus the number of particles, then you get quite high numbers. Typically, we don't want so high numbers. We want something that we have maybe something like 5 per cell or so. The reason why we don't want to have many is because we have to track them during the measurement. And then if you have two particles that comes close, then your measurement setup confuses them and jumps from one to the other. And then you get strange responses in the T1. So you don't want that. But here we were trying to see how many they eat if we want to. Yeah, and then this is like a bit of a summary slide of what we did in that paper. So we have two types of samples here. We have macrophages that have mitochondria in them, and we have isolated mitochondria. And in the macrophage, we worked with two kinds of nanodiamonds. One is bare nanodiamond, and the other kind of nanodiamond is a nanodiamond with an antibody that binds to the surface of the macrophage. So the left particle, the uncoated particle, will be more or less in a random location in the cytosol, while the right nanodiamonds at some point accumulate on the mitochondria. And then we used certain chemicals to trigger a response at the mitochondria. And then what we are trying to see is whether we can trigger metabolism at these mitochondria and see with our diamond sensors whether they respond. Yeah, and the same we also did in the isolated mitochondria. So first of all, I'm showing you the boring stuff, like all the controls, or some of the controls at least. So first of all, how these graphs work. So if you look at the graph on top, on the left axis, we have the relaxation time in microseconds. And on the right axis, we have the radical concentration. So this is estimated from the previous measurements that we did. And then you see the individual experiments. So those are the gray bars here and the gray axis. So each line connects one experiment. So what we did here is we did a measurement. Then we did an intervention, so we added some chemical, and then we did another measurement. And then you see also the colored bars. So those are the averages from all the experiments that we did. So you can do two things. You can look at the statistics of what do cells in general do, and you can look at the individual experiments. What does a specific cell do? And well, if you look at, these are all the controls, as I mentioned before. So what we did here is that we added the particles and we added the chemicals. So to rule out that the chemical itself would cause a response. And we also do experiments where we have the cells, we have the diamonds, but we don't do the intervention. So we more or less just see if the time itself has an influence. So this is the controls that you can see in the bottom row here. So this is when you just leave the cell alone. Well, we are doing a measurement continuously, but we don't add any chemicals. So then you also don't really see any responses. And now this is how these cells look like. So this is what we tried to see here is whether the antibodies that are targeted are really at the mitochondria. This is in this case very difficult to see because the mitochondria here on the left in green, those are kind of everywhere. So it's very difficult to see if they are at the same place. And then what we tried to do is we did some measurements with these particles. So we targeted the particles to the mitochondria, and then we do a measurement before, and then we add some chemicals to see how they respond. And CCCP, so this is a chemical that has a kind of a dose response. So depending on the dose, they do something differently. So in the one case, they kind of shut down the mitochondria, and it's called decoupling. And then you are expecting an increase in the radical response, which is exactly what we see in the middle two experiments here. If you add it in low concentration, it's kind of stressing the cell, but it's not over the threshold that it causes decoupling. So this is the response that we see here on the left. And then the SOD cut here. So this is a kind of an inhibitor. So if in presence of this chemical, we are expecting the radical concentration to go down. And this is also more or less what we see.
May I ask another question?
Yes, please.
So why does covered with antibodies particles accumulate in mitochondria to begin with? Or are they on the surface of mitochondria, or where exactly?
Yeah, the antibody binds to the surface of the mitochondria. So they are more, well, they are not really actively transported there, but they swim around. But when they meet their target, it sticks.
Because mitochondria is quite mobile, right? They're like really traveling and
and
they're basically dragging these nanoparticles with them.
Yeah.
And you can see that in microscopy.
Yes. So I will show a track later on. I mean, it's difficult to see it well because you don't have a very high time resolution here, because we get a location point, something like every 5 seconds. So what we do here at the same time, so we measure the location, and then we measure T1 for 5 seconds, then we measure the location again, then for 5 seconds T1, then the location, and so on. So that later on I will show a track as well. And the experiments below, this is the same chemicals, but in this case, the particles are at a different location. So in this case, the particles are not targeted, so they are somewhere randomly in the cytosol. And in this case, you can see that there is not really the same response that we had in the other case. I mean, they are here at different locations. So some of the particles respond, some of the particles don't. And you can see a very different response depending on where the particles are. Yeah, so this here is an experiment with a standard method. So what we basically get, we also measured here before treatment and the different treatments. And so the difference is that these measurements here, they are from ensembles. So this is not a single cell measurement, but this is like the whole dish. And then you get from all these, you get a measure for the ROS production. But more or less, the trends that you see are similar than what we saw in the relaxometry data before. And then we did try to isolate the mitochondria, so to get data from single mitochondria without their cellular context. And this slide is basically there to show that our mitochondria are mitochondria and that they are still intact. So the left two, so this is the left image, this is a staining for mitochondria, so these green dots that you can see here are the mitochondria. The middle one measures the size, and the right one measures the membrane potential. So this is a dye that measures the membrane potential of mitochondria, and this is basically saying that they are intact. And this is how they look like with the diamond particles. So again, here you see, and now the mitochondria are more spread. So now we can see much better that they co-localize, that they are at the same places. And then we pick some of these particles, and then we did a measurement here. And again, the measurements work similar than before here in these plots. So here again, we have these single experiments, and then the colored bars that are the averages of all the measurements that are here. And then again, you can see that, if you still remember, that the single particles behave here very similar than the mitochondria behaved in the cellular context in terms of this metabolic activity, and that we see more or less the same trends that we saw before. And in this case, the signal that we see is from a particle that sits on a single mitochondrion. And here's the track that I have promised you. This is just one to show you in principle how the tracks look like. In this case, this was a very confined one. So you get, more or less a particle that is zigzagging around this position. So this is here a position around within the cell. And then the color code here is the different T1s that you can see along this track. So what you can do with this kind of tracks is to see whether the track, whether the T1 changes because the particle changes location drastically, or whether it changes because some condition changed. And this you can see very nicely here. So the particle is more or less in the same location, but it has very different T1 values depending on where in the experiment you look. And then I would like to show you a few examples from our clinical work, just to give you a brief look into what's coming soon. So we have one project where we are trying to differentiate between different forms of arthritis. And to do that, we get samples of synovial fluid. So this is fluid that is from the knee joint of patients that undergo a surgery. Then we get the materials, and then we put the material on nanodiamonds. And this you can see on the bottom here. So again, we have T1, and then we have dry. So this is the condition before. Then we add the synovial fluid. Then we see a drop in the radical concentration. And then in the end, we are adding piroxicam. So this is a drug that is given. So it's a drug that has an antioxidant effect, but it's less effective in some types of arthritis than it is in others. And that you can see very well here as well. So if you, on the left side, so this is a type of arthritis that is called osteoarthritis, and on the right, this is called rheumatoid arthritis. And then you can see different responses to this drug depending on what type of arthritis you have. And then another clinical project that we are doing, so this is something where we are soon starting our first clinical trial. So we just got the ethical approval for it. And what we would like to do there is that we look at infertility in sperm cells. And we recently did a study, the pilot for it, the pre-study on boar sperm. And I just show you one figure from this paper. So we looked here in gray. This is if you just measure on the sperm head and don't add any chemicals. And the red curve is if you add a certain medium that is causing these sperm cells to undergo a certain development. It's called capacitation. And this is also a development where they are creating more radicals. And here we are also, in this case, we use two different types of particles. So on the left one, it's oxygen terminated particles. And on the right one is nitrogen terminated. And they are, more or less doing the same thing. So this is just to show you that there is something coming up on sperm cells as well. So that's it, what I wanted to show you today. And at the end, I would like to thank a few people for giving me money, which is all these organizations here. And of course, my team for doing all these measurements and doing all the hard work in the lab. And finally, I would like to thank all of you for your attention.
Thanks a lot for this super interesting talk. I mean, we already had a couple of questions, but I'm sure that there are more. Dima, maybe you go first.
Well, I have two questions. One is, who is this lady in the upper left corner? I'm sorry, I don't recognize her.
That's Rosalind Franklin. That's my...
Rosalind Franklin, of course. Yes, of course. And the other question is maybe more scientific. If you, instead of nanodiamond, you would use like a layer on a plate and just then drop stuff on it. That works just as well, right?
Sorry, can you repeat? You want to put nanodiamonds on the surface and then?
No, I don't want nanodiamonds. For instance, I just want to take a plate, make a layer of these with the same depth.
Yeah.
And then maybe drop take a pipette and drop a whole bunch of different things, and then measure relaxation times in an array like that or something.
Yeah, I would say so. I mean, the only difference is for example, for this measurement in the mitochondria, there you really need to be inside the cell. So that it doesn't work on the surface. But if for the synovial fluid measurements, those are in the fluid, those would be the same, I would assume, on a surface.
Thanks.
There are other questions, thanks, Dima, by Likogos.
Hi, Romana. Can you go to slide 16?
Yes.
Thank you. Okay, so there's one thing I didn't fully understand. These graphs show that the data you see in the macrophages sort of match what you see in isolated mitochondria. But what else? What do you really learn from these plots? Like, what happens? You know, after you put 1 micromolar and then just the before treatment changes, but then slowly sort of increase and then decrease again. What is this plot really?
So what we did here is to compare actually this plot. So this is done with the standard method. And so what we actually compare is we compare this plot with, yeah, oh no, it was wrong direction, with this plot. So this is the same responses that we see here. So the colors are the same. So it's the same chemical causing the same directions of response.
I understand that, but what do those directions suggest about what happens into those systems?
Yeah, so the yellow one, that indicates that you have more radical generation, less radical generation. So in this, the higher, the more radicals. So the same as we saw before. So the yellow one shows that we have less radicals, and in the red you have more radicals.
But the shape is sort of this exponential. Is it expected? Is it linear? Should it be something else? Is the power law? Do you get something out of it, or it doesn't matter at all?
Well, what we did here is to compare it with our measurements.
Okay. Okay. I have 2 short questions. The other one is especially for the arthritis. What could it be an alternative type of measurement? Because I'm sort of thinking like, again, in this case, I think just putting it on a simple diamond works. Nanodiamonds really needed for, let's say, macrophages or mitochondria, which they move around. But in the case of arthritis, just put it on top of a diamond. But
yeah,
other sensors that could work, like organic electronics, like FET transistors, or could something like that work out?
I don't know. It's not really done because those are like very low concentrations. So I think it's not really done, but yeah.
Okay, organic electronics are very, very sensitive.
I mean, what they normally do to do this are they see it in the MRI. They don't really measure the radical load. Okay,
and just one very general question from the first things you said. You said that free radicals are associated with aging. So just a very general question, like how does that compare to the other factors? You know, telomere attrition or whatever. How does that free radicals fit into all the more known and serious reasons why we age?
Yeah, well, I think it's multiple factors, of course. I mean, telomeres is one of them, and radicals is also one of them. I mean,
how do they compare? Like, do we really know? Like telomeres, let's say we have a good theory about it, sort of. But how do free radicals fit into that narrative, and how do they compare?
Well, I mean, one thing that we know about radicals is that they are causing certain damage, and then cells that are damaged, they don't reproduce as much anymore. So you accumulate these kind of damaged cells.
Okay. Okay.
Well, Linus Pauling was a very big proponent of the radical theory, right? That's why he recommended 3 grams of vitamin C a day.
There's another question by Anna Ermakova.
Thank you, Romana.
Hi, thank you for your talk. Can you hear me?
Yes.
Sometimes I have a problem, so maybe I'll switch videos live. I have a few questions. First of all, when you show this tracking and T1 time curve, or not curve, this tracking part, how fast did you perform the tracking as they're focusing for each step, and how did you manage to measure T1 time at this time?
Yeah, so what we do is that we record T1 for 5 seconds, then we do a scan. Yeah, it takes something like a second or so. So we do a very small scan of a very small area with a low number of pixels, then find the new location, go to the new location, and then we do another step of T1.
So it's every 5 seconds.
There are cases when the particles are moving too fast and then we lose them.
Yeah. So and 5 seconds is enough for you to measure T1 time in principle.
Yeah, I mean I mean each sequence you get a value, right? But you are then typically we use a few minutes then for calculating the T1 from those 5 minutes, and a few times in between you did this recentering, right? So you take some values from before the recentering, some after, and then you can you can calculate your T1 from those different measurements.
And also regarding the uptake, when you show so how you, I'm not sure about microfogs, but I think it's similar for all type of cells. When you have endocytosis, initially your nanodiamonds, they're inside of endosomes. And to be attached to mitochondria, they should be somehow taken out of this.
Yeah, so
do you stimulate it somehow, or?
We didn't really stimulate it, so it has been shown that the particles escape the endosomes at some point. But it's very different depending on the cell type. So it doesn't work that beautifully in all cell types. But depending on the cell type, they escape more or less quickly from the endosome, or in some they don't escape at all. So it depends on the cell type, whether they do this or not.
And when you say uncoated nanodiamonds, which kind of surface treatment do you have in this case?
So those are oxygen terminated, so they are made by the manufacturer, and they have oxygen groups on the surface.
Okay. Thank you.
Great, thanks for all the questions. Maybe before you have time. Oh, sorry. Yeah, go ahead.
I'm sorry, I really have to leave because I have to teach in a minute.
All right, Romana, thanks a lot for joining in. This was a really nice talk, super interesting stuff, and you should pop over in Mainz.
Thank you so much, Romana.
Thank you. And if you have further questions, please email me.
We will. Thanks, and see you later.
Thank you.
I just used the dropping out numbers of people to announce next week's heavyweight in the Quantum Seminar. We have Professor Gerhard Rempel from the Max Planck Institute for Quantum Optics talking about quantum networks, triumphs and defeats. So this is definitely going to be interesting. Thanks a lot for